Hello, everyone. My name is Jason Westrich, and I am a postdoctoral scientist in Dr. Elizabeth Ottison's lab in the Department of Microbiology here at UGA. Thanks to UON and the organizers for putting on this great summit. This is the third year that I participated in this summit, and I'm excited to be able to present today. Today I'm going to discuss how our local watershed is a potential hotspot for antibiotic resistance. On the right side of this introduction slide is Brooklyn Creek, which is a beautiful creek that runs through my backyard. It also happens to be one of the most polluted bodies of water in Athens, and during this presentation I will show you a little bit about why this is true. So how do antibiotics and antibiotic-resistant bacteria and genes get into the environment? In this simplified pictorial, the purple arrow shows the flow of antibiotics and the blue arrows show antibiotic-resistant bacteria and genes. As we all know, antibiotics are used to treat a wide range of human and animal infections. Antibiotics are also used heavily in agricultural production to keep animals in modern meat production facilities fat and healthy in these tightly packed production farms. Runoff from these farms is a major source of antibiotic-resistant bacteria and antibiotic-resistant genes. From our antibiotic medicines as well as eating agricultural products, we too are sources of antibiotic-resistant bacteria and genes. Our waste passes through the sanitary sewage system to wastewater treatment facilities, which treat that waste and then release it back into the environment, oftentimes into local bodies of water. Along the way, there's failing infrastructure that accidentally release antibiotic resistance into the environment. For this study, we collected 991 water samples over the course of 5 years, from 2013 to 2018, in conjunction with UON's quarterly samplings. We filtered that water, extracted the DNA from those filters, and then ran quantitative PCR assays for both fecal source markers and AR genes. The fecal sources we looked at were human, poultry or chicken, and ruminant. And as you can see, about half of our samples tested positive for some level of human fecal contamination and or ruminant fecal contamination. A little less so for chicken. We also did quantitative PCR on antibiotic resistance genes for a broad range of antibiotics, including erythromycin, tetracycline, quinolones, and beta-lactams. Some of these antibiotic resistance genes were found in extremely high abundance, including, not surprisingly, some of the older antibiotics like erythromycin and tetracycline, with 74% of our samples testing positive for erythromycin and about half for tetracycline. What we found was all fecal markers, human, chicken, and ruminant, were associated with higher antibiotic-resistant gene abundance. So to walk you through this figure, these are violin plots, which are similar to box plots, but they are a bit more informative in that they show the density of samples at each value. So if that sample is positive for a fecal target, it is represented by the red plots. If it was negative, it is represented by the gray. The number of antibiotic resistance genes is on the vertical y-axis. And what you see here is that the red fecal plots have a high abundance of AR genes, whereas the samples that weren't positive for fecal targets are bell-shaped, showing a greater number of samples with 0 associated AR genes. Similarly, those samples that were positive for any fecal markers, again those bars in red, also were generally associated with a higher diversity of different antibiotic resistance genes. The number of antibiotic resistance genes for each sample type are on the x-axis, so up to 6 different genes per sample that we tested. This is especially true, as you can see, for the samples that were positive for the human fecal marker. As you can see, as you get more positive samples for the human fecal marker, you also see an increase in the different antibiotic resistance genes. It turns out that human fecal contamination is highly correlated with AR gene abundance. This is most apparent when you look at the correlation of individual samples between fecal targets and the number of antibiotic resistance genes. The correlation of ARGs is moderate with poultry and weaker with ruminant fecal markers, but really highly correlated between AR genes and human fecal contamination. with a significant Spearman's rho of 0.6 and Pearson's R of 0.7, showing a high level of correlation. In this figure, we take a closer look at the high correlation of human fecal marker with AR genes in relation to the wastewater treatment. We collected samples from 3 wastewater facilities around Athens over the course of a year. The influent, basically raw sewage coming into the plant, is represented by the brown squares. This influent is treated and then flows back into the environment at effluent sites. We tested this effluent as well, and not surprisingly, you see a shift in the effluent, which is generally lower after treatment for the Bacteroides human fecal marker that we were monitoring, and also some reduction in antibiotic resistance genes. The stream samples in this figure are also colored to distinguish associations with wastewater treatment. The samples in red are geographically downstream from wastewater treatment plant outfalls of treated effluent. What is really surprising in this analysis is the number of no known wastewater outfall samples, the blue dots at the top of the graph, that are extremely high in human fecal input and AR genes, even on the order of raw sewage in some sample sites. This could indicate some major infrastructure failure leading to raw sewage introduction into these streams. During quarterly UON samplings, we typically don't sample every site every time. So in this figure, we are looking at the site-averaged antibiotic resistance gene and human fecal marker abundance, and we again find that they are highly correlated. Looking specifically at where those samples with really high human fecal and AR genes are over the past 5 years, we show some of the usual suspects with those who are familiar with UON data and the impaired streams in the area. One of which being Brooklyn Creek, one of the higher human fecal contaminated sites with AR gene abundance, the one that I showed at the beginning of this slide presentation. Also Tanyard Creek and Trail Creek. I know the county is presently in the process of prioritizing the Brooklyn Creek Sewage Interceptor project for early next spring, and they are already starting to do on-the-ground assessment for this replacement. So that's a ray of hope for ameliorating some of these problems. In this slide, we're looking more broadly across the watershed at specific sampling sites. You see in these violin plots, the more human fecal impact, the redder the plot, and generally the more AR genes you see. Again, dark red Brooklyn Creek, the second from the right, and Tanyard Creek, fourth from the right, as well as Trail Creek dead center are all very red and have high human fecal contamination and a high number of AR genes. So in conclusion, this study has demonstrated that AR genes are widely distributed in the upper Oconee watershed, stream and river samples. AR gene abundance is highly correlated with human fecal markers rather than ruminant or poultry, suggesting that the urban and built environment are a major input source for this contamination over agricultural input, at least in the Athens area. And while wastewater treatment plant outfalls are enriched in AR genes, non-point source human fecal contamination is widespread and a major source of AR genes. So I'd like to finish with acknowledging that this work was funded by the CDC, and also this project has been helped with the close partnership that we have with UON for these many years, as well as the folks at the Athens-Clarke County Public Utilities. And we appreciate ACC for providing these wastewater sewage treatment plant samples. As this was a 5+ year project, there were a lot of hands involved, covering multiple grad student tenures in the Odison lab as well as a few postdocs. I would like to give a special shout out to Julian Damashek, who was a postdoc last year that did a lot of the heavy lifting wrangling this big data set for many of the analysis that you saw today. And with that, I would like to thank you for your attention and a continued great UON Science and Policy meeting. Thank you.